Hello, everyone. My name is Michael Schatz, and I'm your host for today for the Galaxy Training Academy's Meet the Expert series. Our guest today is Dr. Timothy Griffin. Tim's group focus on the development and application of mass spectrometry-based tools to study proteins and the proteomes. The goal of this work is to provide the necessary tools to enable the system-wide characterization of proteins expressed within a cell, tissue, biological fluid, or organism in order to better understand basic mechanisms of biological function and disease. His group also works on the Galaxy for Proteomics, or Galaxy P project, which focuses on multi-omic analysis integrating genomic, proteomic, and metabolomic data to gain new insights into biological system. His work is highly interdisciplinary and collaborative, working across the fields of analytical chemistry, computer science, biochemistry, and beyond. His work with numerous researchers to apply the tools and technologies and problems of biological and biomedical importance. For more information on Galaxy P, and his research team, go to galaxyp.org. Tim, thank you so much for joining us today.
Pleasure to be here. Thanks for having me.
All right, what I'd like to do today is talk first about your background, then we can talk about Galaxy P, and then we can talk about the field at large today. So first of all, how did you get started? Were you always interested in proteomics?
No, I went to graduate school. I'm trained as a chemist and a biochemist. But went to graduate school in chemistry and analytical chemistry. So that's what kind of introduced me into mass spectrometry, the lab I was working in. This was way back in the mid 90s, and sort of mass spectrometry for biomolecules was still in a pretty infant stage. But the lab I was in actually was trying to use mass spectrometry to do DNA sequencing. They thought they
Sure.
could do quick, easy, cheap sequencing, which never really came to fruition for certain genotyping applications and things. It actually had some benefits, but it wasn't then until I went and did a postdoc with sort of one of the pioneers, Rudy Aebersold, when things were really exploding and they were starting to do protein analysis with mass spectrometry, and proteomics was really starting to take off, that I kind of got pulled into that world of proteins and using mass spectrometry to generate lots of big data from big types of biological samples and proteins.
Oh, amazing. It sounds like you're always interested in chemistry and science. I'm just curious, how did that get started?
Yeah, I didn't really know what I wanted to do coming into college. I thought about pre-med or some of those things, but I always had liked sort of chemistry and science, and sort of once I was in college decided I kind of wanted to do more of the basic science part of it rather than kind of go into the medical field and things. So it kind of just led me along. I got pointed in a good direction for a graduate program and a place that would be interesting of kind of, and knew I wanted an interface between sort of analytical chemistry and technology development and something where it had a biological human health outcome. So that's kind of what led me down this road.
Oh, excellent, excellent. In my own work, I typically focus on genomics and DNA. And of course, I'm aware of the central dogma of how DNA is transcribed and then translated into proteins. But I have to admit, I'm pretty novice when it comes to sort of protein and proteomics. Like, what sorts of questions do you ask in the field? What sort of analysis do you do?
Yeah, it goes from somewhat, I guess, basic and simple to much more complicated, partly because proteins are really complicated,
Yeah.
and anybody who's done any sort of delving into protein biochemistry, there's so many facets of what gives a protein function and how do they collectively work together and all of those things. So we do, sort of more standard things would just be sort of quantitative proteomic profiling. It's analogous to doing like gene expression analysis with RNA-seq, where we're just looking at how do certain protein levels change under different conditions. And those are pretty mature technologies and approaches, but then it quickly can get a lot more complicated and maybe more interesting when you start to look at things like post-translational modifications on the proteins. One of the things that we're interested in too is not just sort of looking at what standard known proteins are a part of these systems, but what are proteins that if you're looking at a disease state, cancer or something, and you have mutations or processing changes at the RNA level that might lead to novel different proteins being synthesized and functional, how do you kind of find the needles in the haystack to some degree there and really look at what might be driving disease. So those are at least some of our applications, but it can go from standard to kind of customized pretty quickly, and that's where things get a bit more complicated, and you need tools and all of those things to help you out.
Yeah, I guess I'm quite naive here. I mean, I think of proteins as like little machines that can have lots of, functions and
Mhm.
drive a lot of biological systems. And if I'm hearing you correctly, you might take a healthy tissue, you might take a diseased tissue, and then look at which proteins are specifically in the disease. Is that sort of,
Yeah,
one
sort of
of
looking
the
at dynamic changes and differences. And then, once you get and you're trying to do this at a system-wide level and get a collection of, well, which proteins, yeah, somebody who's been treated in some way versus a control
yeah.
sample, what are the pathways that are changing and things of that nature? The power of proteomics is that you're really looking right at the molecules that are doing the action, they're the enzymes and things that are really the functional parts. So that gets you a little bit closer to some sort of functional change that's potentially occurring in samples. So, yeah, I mean, it's that type of question that many people are asking, and that we're asking ourselves or helping collaborators try to answer.
Yeah, and then my understanding is the mass spec, it gives you a quantitative readout as to which proteins are there and, like you
Mhm.
said, what sort of modifications are there. But there must also be a lot of analysis as to like what the proteins are doing, especially if, I don't know, in a disease state, there are novel proteins that, you haven't seen before. Like what sort of analysis might you do with those?
Yeah, that's a good question. So, to some degree, it is very much discovery science. So, the proteomics platform and the analysis is kind of giving you a readout of saying, well, here are some potentially interesting, even sequence changed proteins. To put any sort of conclusive evidence on, well, what is that protein doing? It almost has to then loop back around to the experimentalist to
Mhm.
say, well, here's what we can show you. There's certain mutations happening. They're linking to some protein change, and hopefully they have a system where maybe they can follow up and do some molecular biology and create the right situation where they knock out that protein or they mutate it or they do something to start to now show, boy, this is a protein that might be driving the proliferation in cancer or whatever it is. So in a way, it's only a starting point to
Yeah.
saying, okay, what's the next question? And there's certainly tools, with AlphaFold and things of those nature where you now can do a little bit more if you change something at a protein's sequence. How does that potentially change structure function? But probably it still comes back to some good old biochemistry, molecular biology to follow up on things.
Yeah. And then in the world of DNA, we have the luxury where we can kind of read off individual nucleotides. We can, GATTACA. We can read out Gattaca. But my
Mhm.
understanding of mass spec is you get sort of an aggregate measurement of mass and charge, and you have to do this matching. I don't know. Could you walk us through like how that process works?
Yeah, the predominant way it's done is we don't actually analyze the intact proteins. We, take a mixture of proteins and we actually purposely break them into peptides. So we use an enzyme that chews them up into smaller peptides. That's what the mass spectrometer is able to then take mixtures of peptides, and it ultimately is trying to tell us what is the sequence of all the peptides that it can detect in this sample. And then there's some bioinformatics to say, let's go back to a database of known or predicted proteins and match those peptide sequences to those proteins. So it never really gives you a full, I mean, there's an analogy to sort of long range versus, or long read, RNA sequencing versus short read. We're
Yeah.
generally getting short reads on these proteins, and we try to, piece back together the jigsaw puzzle and infer proteins. But it definitely gives you powerful information as to we found all these peptides that map to this protein. So this protein is present and it changes in its abundance or whatever. But it does also has some limitations that we don't get just a full-on readout of, from end to end of the amino acid sequence. It's a little different the way the platform works that way.
And then in round numbers, like what's the complexity? Is if I give you a sample, how long does it take to prep it? How long does the
Yeah.
instrument run? And then how long does it take to do the analysis? Again, I'm very
Yeah, so those are relatively mature processes
Yeah.
now. You take, depending on the question, but standard thing is you take tissue or cells, and there's ways to homogenize and solubilize, and you extract all the proteins out, get those processed and push them through the mass spectrometers. The mass specs are becoming more automated. So, you maybe take a day or two to prep a sample or prep a large group of samples, put those in an auto sampler and let the mass spectrometer run those through. Depending on the platform, it may be an hour a sample, but there's been changes now where that is accelerating and they're getting down to under a half an hour or 20 minutes and more sensitivity and all of those things. So it can take, a matter of weeks or so to get a pretty full view of a set of samples and what is the, proteome dynamics.
And what are some of the pitfalls, both experimental and computational? What are some of the challenges you might encounter?
Yeah, there's plenty. We're still, in terms of the analytical piece of it, and very much improving, but we're still somewhat limited just by sensitivity of the mass spectrometers. It's always been a part of this where you have a really large dynamic range of proteins abundance in any given sample, where you've got some proteins are in there at tens of millions of copies, and then you got other proteins that are maybe really important, but they're at a few thousand copies per cell. So this really big difference, and we don't have PCR for proteins. We can't just amplify signals. So there's always been, how do you dig down in and find those low abundance things? Getting better, but that still can be a pitfall of just you can't find some things, or certain sequences just don't get ionized and fly well in a mass spectrometer, so you don't detect them. So still always some challenges there, because if you don't see something in your sample, it doesn't necessarily mean it's not there. It means we just don't detect it. Um, and at I guess the analysis or the data analysis level, there's some pretty mature, if you're just trying to do standard proteomics and quantify the differences between samples, pretty good array of software options. Scalability is one that, as these instruments get better, people now are be able to say, well, let's do hundreds to thousands of different patient samples, which before would have been a really big task for proteomics. At least at the analytical side, you can actually do that and generate the data, but that now is starting to present scalable software to handle, large numbers of samples, and how do you push
Yeah.
those things through and how do you manage that data? So that's becoming one of the challenges. And other things, depending on the question. If you are really trying to find novel sequences that map back to some mutation at the DNA level, trying to control for false positives and making sure you're really accurate and cautious in what you're finding, and are these real hits or are they some other artifact in the data? There's lots and lots of potential pitfalls in there as well.
On, say, I don't know, genome or gene sequencing or transcriptome sequencing, there's at this point there's pretty well established tools for like QC of the data,
Hmm.
sort of a primary analysis where you align or maybe assemble, kind of secondary analysis where, I don't know, you're doing differential expression, do some statistics. What sort of tools and file formats are most relevant for mass spec?
Yeah, a good thing in the mass spec world is, a while ago they kind of came together, and there's different vendors that make different platforms and they have their own file formats, but they kind of came to the agreement that we should be able to convert these to like generic file
Yeah.
formats. So we can do that, which is great, because that then gives us the chance to develop software tools that can find, analyze data from a variety of instruments, and just makes things much more interoperable. Most of the programs that are pretty mature now have ways to take the data and do some quality control. Like, they estimate false discovery rates when it comes to, like, when you're trying to identify these peptide sequences and then infer the proteins. They can give you an output in a report that says, to our best ability, we think this whole data set is maybe at a 1% false discovery rate. So you can be pretty confident that what you're seeing is real. And then from there are some packages also and it becomes a little bit more generic, just like, gene expression analysis. Now you have proteins and quantitative values, and there are some specialized statistical packages that you can plug that into, but it becomes kind of a similar problem of, okay, here's quantitative data. How do we statistically analyze it and determine things that are significant? And so that, there's a variety of tools that can do that as well.
Okay. What I'm hearing is, the file formats now are, I don't know, standardizing, but then there's a variety of tools. You have to build workflows. So maybe this is a good
Mhm.
segue. Tell us a little bit about Galaxy P.
Yeah. Yeah, so as you picked up, I didn't come into this as a computational biologist. I had only a little training kind of in that area. I think it was just always a part of the work was always, hey, we've focused on these instruments and how do you prep samples the right way to optimize what the instrumentation can do. And there was always a lot of data getting put out. So I worked with informaticians to help generate some, early data software that could take some of this data and do something with it. And after I was at Minnesota, boy, 20 plus years ago now, we started generating our own data and started mostly kind of some of these questions started coming up like, hey, we have, say, RNA-seq, or we have some sort of sequencing data that's predicting from that novel protein sequences might be expressed in this sample. We have the mass spectrometry data that we want to match to that, but suddenly there wasn't really a lot of tools out there to do things like that. It was beyond what the standard things were, not geared to do that. So that's where we started to kind of work on campus. We work with the Supercomputing Institute, the Minnesota Supercomputing Institute, and sort of were saying, we need some sort of platform, do we need to develop our own software, or could we use certain other software that could get kind of linked together? That led us to Galaxy. They were already kind of looking at Galaxy as a way to do genomic analysis and said, it's kind of this open platform. Why don't we think about proteomic kind of integrated workflows? And the fact it was already on the genomic side, pretty mature and really had a lot of tools, we started to then pull in certain appropriate publicly available software to do the proteomics and then kind of develop the bridging tools to say, let's bring these things together. So it really was driven by we need to do things in a more multi-omic way. What's a platform that can do this without wanting to reinvent the wheel and try to develop our whole own platform? So it was really a nice option that we explored, and the rest is history to some degree. So,
I mean, it sounds like a success story for open science and
mhm.
open source software. We could take an existing platform developed in one context and apply it in an entirely new context.
Yeah.
And then in kind of like round numbers, like you said it's been up and running for 20 years. Like, I don't know, what kind of projects, what kind of
Mhm.
usage have you seen of Galaxy P?
Yeah, it's probably one of the things that's a little hard to track because of the fact that it's so open and distributed out there that we always kind of say, we definitely have developed certain specialized tools that we can go in and look at how many downloads, thousands and thousands of times people are
Yeah.
using these. So that gives us some measure. But, we personally have kind of used the platform as we've developed it to kind of apply it across a whole number of different samples or projects. Kind of over the years, we have a lot of cancer focus. So we boy, have worked with numerous groups, both here at Minnesota, but outside where, they have a question, they have the right system, and they're already wanting to generate some sort of proteomics data and or multi-omic data, some RNA-seq with that. So we've kind of partnered with them to say we've got the tools to do this. So a lot of those things, one of the interesting early on projects, we partnered with somebody here that does ground squirrel hibernation. So
Huh.
Minnesota is the gopher state, and the ground squirrel, the 16 lined, I think, ground squirrel is the gopher, common gopher. And they studied how it is gophers go from this dormant state in the winter when they're hibernating, and they will have these quick bouts where they'll wake up and really quickly jump awake, and then they go back to sleep. And they were trying to study like how does their cardiovascular system and things kind of do that, because you have to be so elastic and able to do that. So we actually did a RNA-seq and proteomic data study where they were looking at how does the gene expression change, but then how does that also affect proteins. So we've had some interesting times with it, I think. Found some
I like
interesting projects.
I like the range from, piercing into human health and cancer, but then also, the sort of basic biology and
Mhm.
metabolism of many other species. It sounds like there's an endless set of questions that you could address here.
Certainly is. Certainly is. It does span generally spans lots of different projects that are doing that type of biochemistry. So,
And then in a more, I don't know, technical level, are there any like features or capabilities of Galaxy that you really like, or your users really like?
oh yeah, definitely. I mean, it really gave us an in because, like I said, I wasn't coming at this with a real advanced computational expertise or lab, so it fit really nicely because of how it was already designed to be usable by everywhere from people who are more advanced bioinformaticians and computational biologists, but to those who are pretty novice, and you
Yeah.
could jump into it pretty easily. So that part I certainly like, and I've said this before, it's easy enough for a professor to use. So I was able to learn it pretty quickly, and I do little dabbling on the sides and help push along projects, and it with a little bit of learning, it's usable, and I can tweak this and change that and see how it, changes results. We've had amazingly good success also training undergraduates who come into the lab and, have very little experience, but you put them on some of the training material, and next thing you know, they're running their own analyses and doing work. So, that for me has been really the one of the biggest things is just the accessibility and the usability for anybody to use this. And then I guess the other is just the ability to really pull in different tools and bridge them together and make them interoperable into a workflow has been huge because that's really what we've needed in our work.
Yeah, I mean, I agree with everything you just said, right? It lets you really focus on the science in that, you have your toolbox, you have all the tools you might need, and you can just immediately start running with it. I mean, behind the scenes, it's, state-of-the-art scientific software, but just sort of takes out all that overhead of getting them installed and making sure they
Right.
work and integrated. And then, like you said, once the tools are there, you can build really sophisticated workflows.
Hmm.
Are there any, I don't know, do you have any favorite tips for the audience today? I don't know, some special feature or Galaxy that you really like that maybe isn't well known?
Boy, I don't know if it's not well known, but we try to make it more and more known, and that's just the whole training network is what is such an awesome feature because you can
Yeah.
just what a resource to just be able to not just point somebody to here's a document to go read and you have to follow it and how do you analyze your data and all that, but an actual hands-on interactive here's your data, use it, follow these instructions and actually build your own workflow that pretty easily can get then translated to analyzing your own data. So I mean, that to me is still the I think the most unique and powerful part about Galaxy. There's a lot of powerful pieces to it, but that ability to point people to things and let them learn on their own without, them just getting lost is so huge. And we use it all the time. We contribute to it all the time. So I mean, that's the piece that I think I wish more people knew about because I think it's so powerful.
Yeah. I mean, that's the theme of this whole week for the Galaxy Trading Academy is, expose people to all the trainings that are available through
Hmm.
the
Hmm.
GPN. But like you said, these are not static resources. People
Yeah.
can contribute to them, grow them as new tools come online, new topics come online. We
Yeah,
can we
yeah.
can
Yeah,
contribute.
I mean, and if I had a kind of a tool thing that I could would point to, it's what Galaxy is really great for is taking outputs, and it's really been great for what we do because you get outputs from some software that immediately need to get reformatted into
Yeah.
some, other tabular file that's slightly different. Those tools are great because it just automates that whole process. You're not going into Excel and trying to do it yourself and then re-upload. So we've definitely leaned on those heavily to make our workflows kind of interoperate and automate things. Those are nice tools at that level.
Fantastic. So if we could shift gears just a little bit, I'd like to talk a little bit about maybe the future of the field. I mean, you already
Mhm.
mentioned AlphaFold. I mean, are you seeing widespread adoption of that in proteomics?
Yeah, I think more and more, and we haven't done a lot ourselves, but it suddenly is this tool that's there that's usable and should be kind of, hey, because that's like we were saying, a lot of times the outcomes of these proteomic analyses is, all right, I got my list of proteins, maybe
Yeah.
I've got my list of changes to certain amino acids and proteins. And the question now is like, well, now what? Or so what? What do I do with this, right? So those types of, deep learning type of tools that can help you interpret what at least might be going on is, I think, definitely something that needs to be more kind of integrated in. And I know Galaxy has that opportunity to do that, and at least just place in the hands of the experimentalists then some more directions to go potentially. If they could have a readout that says, okay, here's the proteins that you should be interested in. Here's a further analysis of maybe the functional changes that those are a part of, and give them a way to really prioritize what they want to do next potentially.
Yeah. And I see this like rise in, what you said is multi-omics, where it's, kind of multiple assays. Like
Mhm.
what's the advantage in your mind to that sort of information?
Yeah, I mean it's critical because, there's more and more that just says if you look at those, domains on their own, they're interesting. You see some gene expression changes, and you can look at pathways, you can look at proteins, you can look at pathways, but most likely what is driving so many things is there's this interconnectivity between, rare transcripts that are producing some rare protein that has some slight structure function change to it. So without being able to at least predict what those changes are and connect it to the proteins and things like that, you probably never see them. And so
Yeah.
you won't know there's this underlying biology that's really going on. So more and more to me, just looking at, being exposed to certain research and things, it's that interface between sort of translation of RNA to proteins, and there's probably a whole sea of very novel proteins floating around that are, again, kind of the needle in the haystack that we don't know about, but how do we connect the data to start to at least give us some notions at what's there? So, it seems to be the way this has to all go, and then it all relates back to the genome too, because that's where so much of this is, these tweaks are happening at the genome level. That's then this cascade of other changes.
Yeah, and on the experimental side, I've been hearing, about a variety of mostly startup companies proposing like nanopore-based protein sequencers. What,
Hmm.
what do you think? Is that going to come true? And how might that
Yeah,
change the
I
field?
think it is. It's maturing. And for certain applications and certain questions where, yeah, we really do need, because like I said, one of the limitations with the mass spec is it can do lots and lots of things, but it isn't really a true protein sequencer. So it
Right.
doesn't take it end to end and say, here is exactly your amino acid sequence. So some of these more high throughput, single cell, maybe whatever it is, ways to sort of take proteins that could be disease related and actually at the amino acid level, give you a readout. I think it certainly has its place. I think all of these are complement each other, so I don't
Yeah.
think it necessarily takes out of the play using a mass spectrometer for sort of global protein profiling, but definitely has its place. And they seem to be coming along. There's always this big burst of excitement, but then this lag of let's get the technology to a place where it's really robust and usable and all of that. And I think they're still in that phase, but
Yeah, it feels analogous. I was one of the early access customers on the DNA nanopore sequencers, and those early days were rough.
hmm
But here it is, I don't know, 10 years later, and now it's quite mature and you can do
hmm
amazing things with it. So, over I don't know, 10 or 20 year horizon, the sky's the limit of what's possible.
Yeah,
But if I
definitely.
hear you, I mean, it kind of spans from, both the experimental sciences and the computational sciences. You mentioned kind of the rise of AI. Are there any other kind of, I don't know, experimental or computational techniques that you're excited about?
Yeah, I mean, kind of like I alluded to this, that there's been in the last several years a real big jump in some of the mass spectrometry instrumentation, so that, like I said, the instruments incrementally have always been getting better in terms of their sensitivity and their throughput, so you could do more samples in less time, but also then dig deeper into like a proteome and what you can detect. There's been this pretty big leap where we now have some mass spectrometers where, we're doing in 20, 30 minutes what would have taken weeks to months before in order to get depth.
Yeah.
So Those are great because now you can start to do cohorts of samples that are larger. You can and not sacrifice depth of information. If anything, you're getting an order of magnitude more depth, so you can really kind of cover a proteome. So that's exciting. It's also bringing up, more data, deeper data. You need software tools, and if nothing else, just systems that can manage that amount of data. So I think Galaxy really has a place there to be able to bring in these larger data sets and manage them and put the tools to them. And ultimately, there's always emerging, deep learning tools that are helping us better analyze that data as well. So Galaxy is again, sort of a perfect collection point for all of those things.
So it's making, I don't know, today's problems faster and more efficient, but at the same time opens up entirely new research
Yeah,
questions.
suddenly things you got to think about of, well, we never had this much data, and how do we just manage this data and analyze it correctly and things like that.
All right. So, but it sounds like it'll be a long time investment into the future to become proficient with Galaxy today, so that in 20 and 50 years,
There
it'll
you
be
go.
all the more efficient for them.
Sure.
Awesome. 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 Well, just one last sort of major question for you on the training academy today and this whole week. It spans from expert users to novices. So if you're a novice getting started, entering the field, but you aren't really sure where to get or how to get started,
Mhm.
what are some of the main tips you have for them?
Um, first thing is that, when you go in and try these, I tell this to all the beginning students that we have, you're not really going to break anything. So make mistakes. That's how it
Yeah.
always goes. You make a mistake, you figure out why, and you correct it, and that's how you learn. As I said with the training academy, what's really great is that they have the very basic introduction to Galaxy trainings, which someone doesn't really have. You don't have to be a computer science whiz to do this. It's very achievable. You go in there and you follow it, you get comfortable, and then next thing you know, you build on that and you're get to become a more sophisticated user, and maybe you're developing your own workflow and inserting certain tools and doing things like that. So play around with it. It's kind of fun too because you see it run and you get results, and those results are interesting, and then that leads you kind of sort of delving into, well, what protein did I find? What is this, and what's interesting about it? So yeah, I don't know. It feels like to me kind of a sandbox to almost start playing in, and all of the resources are there to get you started.
I really like that sentiment. I think at the bench, there's a culture of experimentation and trying, different protocols. And I think we can sometimes get locked into, oh, it's on a computer, so you must do this, must do this. But I really
Mm-hmm.
like that sentiment of, let's experiment. Let's see
Yeah.
what happens when we try this. Let's see what happens.
Yeah.
You're in
It's
this
built
really
very well for that. You can just rerun what you did and change
exactly,
this or that, and what
exactly,
happens now?
exactly. I really like that sentiment of, that sandbox where you can be free to experiment without worry. I really like it.
Hmm.
Well, thank you so much for all your time today. This has been a really exciting conversation. Again, if you have questions, if you have uses for proteomics, go check out galaxyp.org. Thank you very much, Tim.
Good thing. Thanks, Mike.
Have a great day. Bye now.